So today, I actually thought I'd also broaden out. I'll talk about our work in the Brassicaceae and Arabidopsis relatives, but also some other systems as well. And so in particular, I want to talk about adaptation and constraint in plant genomes. Or actually, that's a bit of false advertising. Let's see if I can. So in fact, a lot of our work and a lot of our interest is sort of the opposite, which is maladaptation and loss of constraint. So to what extent do essentially we have borrowing a phrase from Bill Rice, do we have cases where evolution goes backwards? So I put up one of the classic well-known examples where evolution goes backwards. And again, Bill Rice used this phrase to describe Y chromosome degeneration. So we have the human X and Y chromosomes where the Y chromosome is a highly degenerate version of the X. Okay? So this is a case where we have widespread degenerative evolution, where large-scale regions that used to have gene function have now become degenerate and lost. So why does this happen? Why do we get backwards evolution? Another example of this occurs very often in plant evolution. So when we look across angiosperms, the stars here, the green sunshines here indicate cases where plant genome evolution has undergone whole genome duplication. So plants have experienced polyploidization events. But over time, what happens is you get a diploidization process where many of these genes that were duplicated at some point end up becoming degenerated and lost. Okay? So again, we have this process of backwards or degenerative evolution. Large-scale regions of the genome that used to function are no longer functioning and have become inactivated. Okay? All right, so in many ways, this is sort of counterintuitive. Why should evolution proceed in this backwards direction where you get sort of loss of constraint? Now, some people might argue, well, if we think about males, maybe thinking of them as having a degenerative chromosome where they've evolved backwards may not be so surprising, but I would still attest that it is somewhat counterintuitive. So natural selection should act to improve function. So why does a loss of function occur? How do we get these sorts of events? So one of the recurring themes here with things like sex chromosome evolution, as well as whole genome duplication, is you had genes that used to be interacting with each other through recombination, and that interaction has been shut off. Right? Y chromosomes become non-recombining, or through whole genome duplication, you have duplicate genes that used to recombine with each other and are now no longer interacting with each other. So gene copies that originally shared the same evolutionary history are now evolving independently. Okay? So if we really want to understand how and why this occurs, we have a sort of challenge. So the human Y chromosome is highly degenerate. This is a very ancient event. So if we really want to understand how and why this happens, it's pretty hard because we're sort of at the end point of degenerative evolution in the mammalian Y chromosome. Similarly, if you look at the Arabidopsis genome on the right, there's a footprint of very ancient events of whole genome duplication, but it's clearly extremely ancient here. And so really understanding the early stages and how and why this occurs is quite difficult in that context. And so I would argue that plants actually provide a very useful system for understanding many of these processes of degenerative evolution. And studying the population genomics of plants is quite useful in this context. And that's because they're routinely going through transitions in polyploidy or mating system shifts. So there are many young Y chromosome systems in plants, for example. There are many recent mating system transitions and very recent whole genome duplication events. And so one thing we're doing in my lab is really focusing on the population genomics of these early transitions in order to understand better the how and why these transitions occur and genome evolution, the consequences for genome evolution. Okay. All right, so we can think of various possible explanations for this degenerative evolution. So one of the most intuitive, I think many of you may think about, and many of us sort of assume is sort of the null model, is this is a passive consequence of gene redundancy. We have a copy on the X. Why do we need a copy on the Y? So it may just sort of be a passive neutral degenerative process. However, that's probably not the only explanation because most mutations, most deleterious mutations are not entirely recessive. So they may not be completely masked by the other copy. So another major player may be the role of inefficient selection. So there are various reasons why selection may be inefficient in these contexts, and I'll talk about how and why that might be. And then the other possibility is you have differential adaptation. You have two copies of the gene, one of which is carrying on adapting, the other is not, and then that might set off a process even of adaptive shutoff of these gene copies. Okay. Right, so why would selection be inefficient? Well, there are a number of sort of population genetic models that would predict why selection might, in some cases, be less efficient. So small effective population sizes, if you have a small number of copies, that can lead to the dominance of drift over selection. If you lose sexual reproduction or recombination, that can lead to inefficient selection. And higher ploidy levels or gene copies. And I'm going to focus in particular on these two in the context of what I want to tell you about. So the loss of recombination or low levels of recombination leads to less efficient selection. And here I'm showing this in the case of negative selection, but it applies to positive or beneficial mutations as well. So if you have a region of high recombination and many deleterious mutations in this population, well, through recombination, you can generate mutation-free chromosomes. In contrast, if you have a region of low recombination, essentially only one of these chromosomes is going to persist, but you can't recreate a mutation-free chromosome. And so you end up with fixing many more deleterious mutations in the absence of recombination. Okay? And so this is thought to have wide-ranging implications, for example, for the degeneration of sex chromosomes, for understanding why sex is so popular out there in nature, among other reasons, and also the evolutionary maintenance of outcrossing. So both sexual reproduction and outcrossing may be maintained because selection becomes more efficient with high levels of recombination. So we've been quite interested in this in the context of mating system evolution from outcrossing to selfing. So this is one of the most recurrent evolutionary transitions. It's one of the most striking examples of parallel evolution, I think, in the biological world, where you have these recurrent shifts from large-flowered outcrossing plants to small-flowered selfing plants. You get this parallel morphological evolution where the plants no longer are investing in attracting pollinators. And it's also a very important mode of speciation in plants. So why does it evolve? Well, there are a number of theories. One is there's a transmission advantage. So if you're both mother and father to your offspring, you have a transmission advantage in the population. And also, if there's no other mates or pollinators around, you can mate with yourself. Okay? So in those contexts, selfing will be favored, even though there may be the harmful effects of inbreeding oppression. Okay? However, even though it might be favored on the short term, there's this long-standing hypothesis, originally promoted by Stebbins, which is that these transitions are, over the longer term, an evolutionary dead end, because losing recombination, because you're highly homozygous, will essentially limit adaptive potential and lead to mutational load. However, there's been pretty limited evidence. So there's been some phylogenetic patterns fitting with that, but there's somewhat limited evidence that selfing really does lead to an accumulation of deleterious mutations. And so as part of a big collaborative effort with the JGI, with Jeremy Schmutz and Dan Roxar, and in collaboration with Detlef Weigel's group and my former postdoc, Tanya Slata, we sequenced the genome of Capsella rubella. So this is a very close relative of Arabidopsis. And one of the interests here is that Capsella rubella has very recently evolved this transition to selfing from its outcrossing progenitor, Capsella grandiflora, and rapidly spread in blue here throughout Europe, whereas Capsella grandiflora in purple is restricted in range to Greece, to a glacial refugium. So having whole genome assembly and population genomic data allows us to really get a glimpse at the earliest stages of genome evolution following the transition to selfing to address this issue about whether this transition is actually an evolutionary dead end. So one thing we see when we look at patterns of genome-wide diversity along chromosomes, in black you see the polymorphisms that are unique to Capsella grandiflora. In blue you see shared polymorphisms between the selfer and the outcrosser. And in red you see polymorphisms that are unique to the selfer. So there's a genome-wide collapse of genomic diversity that's almost complete associated with this transition to selfing. So both the founder event and the switch to selfing has led to a severe loss of genetic variation. There are some regions of the genome where there's been more shared variation maintained. And in fact, this is associated with regions of where there are disease resistance genes. So this may be the maintenance of balanced polymorphism. But in general, we have a genome-wide collapse of genetic variation. And when we quantify the strength of selection genome-wide, we see a consistent loss of selection in the selfing genome. So here, what we're estimating here is the proportion of sites that are evolving non-neutrally, so in other words, the amount of constraint, in protein coding regions, non-coding regions as a whole, and in conserved non-coding regions. And in all cases, the selfer shows a consistent weakening of the efficacy of selection. So we're really seeing, even in the early stages, within about 100,000 years of evolution, a rapid decline in what we're interpreting as a decline in fitness and evidence for an increased mutational load. And this seems to be ongoing. So when we look at the ratio of non-synonymous to synonymous polymorphism, it's highly elevated at polymorphisms that are unique to rubella. So since the transition, there's still an accumulation of deleterious mutations here. Now we could imagine a couple alternative explanations here. One is we hypothesize. This reflects inefficient selection in the selfing species. But another possibility is maybe there's a lot of relaxed constraint, getting back to this idea that it's a passive process of degenerative evolution. So half the function of flowers is sort of gone in a selfer. There's no longer investment in outcrossing. And so if a large number of genes are associated with investment in outcrossing, there may be relaxed constraint. And maybe population expansion has also led to relaxed constraint. So one way to get at this is to look at genes that are differentially expressed between the selfer and outcrosser. We see a bunch of genes that are downregulated in the selfer. Those are associated with things like pollen production because selfers are sort of abandoning investment in male function related to attraction and outcrossing. And then we see some upregulated genes, which interestingly are more related to female function. But in both of those cases, as well as genome-wide, we see the same signal of relaxed selection. And similarly, when we model the process of the transition to selfing, we can basically explain the patterns of diversity we see at non-synonymous and synonymous sites just through the transition to selfing and its effect on selection. And so overall, we can explain the patterns primarily by this idea of inefficient selection rather than this idea of relaxed constraint. Okay. All right, so overall, we see evidence of inefficient selection. However, in a selfer, this inefficient selection shouldn't be complete, and we certainly don't see signs of widespread degenerative evolution like on the Y chromosome. This is more subtle, relaxed selection. And one way to think about this is, well, selfers are highly homozygous. And so we still should have purging of strongly deleterious recessive mutations going on in highly selfing populations. And so this brings up the sort of other factor that's likely playing a very important role in degenerative evolution and inefficient selection, and that's ploidy. Okay, so if we have a haploid or homozygous condition, and we have deleterious mutation or a beneficial mutation, they're going to be seen by selection, right? So a strongly deleterious mutation will be eliminated if it's haploid. A beneficial mutation will spread. However, in the case of diploid or heterozygous states, or if we have a polyploid, for example, you can get greater masking of these deleterious mutations, and they're more likely to persist, and beneficial mutations are more likely to be lost. Okay, so higher ploidy can also weaken the efficacy of natural selection. And a grad student of mine, Ramesh Arunkumar, looked at this in the context of Capsella grandiflora data, where he was comparing genes that are expressed in the pollen, so that would be in the haploid phase, to genes that are not. And he found a consistently more efficient selection, more evidence for strong purifying selection, and beneficial mutations in genes that are expressed in pollen compared to those that weren't. So that's consistent with the idea that ploidy also plays an important role here. And so recently, Gavin Douglas and colleagues were looking at another member of the Capsella genus, Capsella bursipastoris. So this is a highly successful worldwide weed, which has experienced a recent transition not just to selfing, but also to polyploidy. And in fact, we see a greater, more severe evidence of the accumulation of deleterious mutations in this system than we see in Capsella rubella. And so when we have ploidy combined with the transition to selfing, and through simulations we could show that both are playing an important role, indeed we see more evidence of recent rapid degenerative evolution. All right, so now I want to shift gears, again, thinking about now when we start thinking about the combined effects of recombination and ploidy to plants and sex chromosomes. Okay? So plants, a number of plant species do have sex chromosomes, although as I mentioned, they tend to be much younger in age than mammalian sex chromosomes. And that allows us to look in more detail at the earliest stages. So one of the most well-known plants with sex chromosomes would be hemp, for example. But Rumex and Silene are additional species that have recently evolved sex chromosome systems in plants. All right, so again, we can sort of return to some of these questions. Is most Y chromosome degeneration passive? For example, maybe dosage compensation happens early across most of the chromosome, and then we get neutral accumulation of deleterious mutations on the Y chromosome. Does a Y chromosome degeneration proceed, in contrast, because of inefficient selection, because there's a loss of recombination? And it's also been hypothesized related to this idea of pollen expression that actually in plants, Y chromosome degeneration may not occur or may not occur to such an extent because there's widespread expression of genes in the pollen phase, in the haploid phase. Okay, so is it easier for a Y chromosome to degenerate in mammals because it's permanently heterozygous than in plants because many genes will be expressed in this haploid pollen phase in the case of plants? Okay, so now we're starting to think about this interaction between recombination and ploidy here. So to look at this, we used a model system, Rumex hastatulus. So again, this is a dioecious system where we have separate sexes. The sex chromosomes have been shown to be heteromorphic and somewhat heterochromatic on the Y. But what's very interesting about this system is that there's two races. There's one that just has one Y chromosome and one that has two Y chromosomes that's been very recently, over the last 20,000 years, evolved from via an X autosome fusion. So the interesting context here of these two races is we can start investigating the time scale in which we get this degenerative evolution over this very recent time scale, in addition to this older Y chromosome system. So that can allow us to start asking what processes are happening earlier and later in this process of Y degeneration. So Josh Huff, a grad student working with Spencer Barrett and I, conducted a transcriptome-based identification of sex-linked genes. So he did a cross, and we sequenced at the transcriptome level a number of males and females. And currently, we're doing genomic work along similar lines, looked at segregation patterns to identify both sex-linked genes where there's an X and Y copy, as well as hemizygous genes. And so in the XY race in that system, Josh found, for example, the brackets, it depends on your level of stringency for what you're calling sex-linked genes, but about 700 sex-linked genes with Y copies and about 119 genes that showed hemizygous segregation patterns. Keep in mind, currently this is at the transcriptome level, so we can't distinguish gene silencing on the X from actual deletion, but that's what our genomic work right now will lead to. And so Josh inferred about 24% of the genes were hemizygous. So this implies pretty extensive gene silencing and loss already in this relatively young sex chromosome system. And we saw similar patterns for the 2Y race for genes that were shared with the XY system. Now, the XYY unique genes, these should be the very youngest within the last 100,000 years of evolution. And already, so we see a lower percentage of genes that show hemizygous patterns, but already there's signs of degenerative evolution there. About 8% of genes show degenerative evolution. In addition, we see a sort of age-dependent pattern of gene degeneration in terms of relaxed selection. So we see a strong signal of a large number of non-synonymous relative to synonymous changes on the old Y, and less of that pattern, but still more evidence of relaxed selection as well on the young Y. Okay, so even at the very earliest stages, we're seeing signals of relaxed selection, deleterious mutation accumulation on the old and young Y chromosome. We're also seeing a reduction in gene expression that's age-dependent on the Y chromosome. But what we don't see is evidence for dosage compensation at these hemizygous genes. So if we look at expression levels in females compared to males, the hemizygous genes show about 50% expression in the males. And so if we think about the timing of events here, in the context of this plant anyway, dosage compensation does not precede relaxed selection. And so this implies it's the loss of recombination that's really limiting or weakening selection. And we don't have already a sort of passive process because expression has already evolved here. Okay? And so thinking about what this might imply about haploid gene expression, it implies the loss of recombination might dominate even though some of these genes may be expressed in the pollen phase. And in fact, in Spencer's lab, there's evidence that Y-bearing pollen may be less competitive, so actually having experienced either genetic load or less adaptation. And so if you look at the relationship between how much pollen you put on a stigma and the sex ratio, you see the plants get more and more female biased as you add more pollen, implying the X-bearing pollen is more competitive than the Y-bearing pollen. And so taken with the evidence from the genomic data so far, this implies that this degenerative evolution may be outweighing essentially the Y may be becoming less adapted than the X, and this actually has consequences for things like the sex ratio. Okay, so finally, so now if we think about losing recombination and permanent heterozygosity, the perfect storm for this degenerative evolution is the loss of sex entirely. So if we have a diploid that's lost sex, we now have the entire genome as permanently heterozygous and permanently lost recombination. So this would be a really interesting system in order to investigate degenerative evolution. But to do that most powerfully, of course, ideally, we would have replicate species pairs across different timescales. And then we could investigate the role of timescale in this accumulation of deleterious mutations. So to look at this, the Oenothera system, the evening primrose system, actually provides a really nice model in order to look at this. So we have a number of replicate transitions from sex to asexual reproduction. I'll explain why I have these in quotation marks in a minute, where the red indicates transitions to asexual from sexual reproduction. So about 30% of Oenothera species have what's called permanent translocation heterozygosity. They've completely abandoned effectively sexual reproduction. And this has occurred many times independently. Okay, so what do I mean by effectively asexual? So in the sexual evening primrose, we have normal segregation. So if A, B, and C are different chromosomes, we have in the pollen and ovules, we have normal segregation of these chromosomes. You can get different combinations in the progeny. Now, in species that have this permanent translocation heterozygosity, the pollen will have one of these haplotypes, the ovules will have another, and there's absolutely no segregation across chromosomes occurring as well. So no recombination and no segregation. And through self-fertilization, essentially this is maintained intact. So these permanent translocation heterozygotes are functionally asexual. The reason why this happens is essentially there's been translocations throughout the genome. And these species, as their name implies, are permanently heterozygous for these translocations. So segregation doesn't work right. You get this meiotic ring formation. And essentially, half of the genome goes to one gamete and half to the other. And you get no segregation and recombination. And that's coupled with balanced lethal mutations, such that essentially there's a pollen genome and a maternal genome, an ovule genome, and nothing in between. So you sort of feel bad for Hugo de Vries trying to rediscover Mendel's laws with this system. And again, self-fertilization preserves these lineages. So very quickly, we examined, so Jesse Hollister, a postdoc working with Mark Johnson and I, looked at, again, we started with transcriptome, and now we're going genomic. Looked at transcriptome data from 30 species, representing 10 independent transitions from sexual to asexual reproduction. The first thing we see is, indeed, there's higher levels of heterozygosity in these asexual populations compared to the sexuals. And that's because, again, you have these permanent cleavage of interactions between the genomes. And so over time, you get higher and higher levels of heterozygosity. And in addition, we see an age-dependent evidence of accumulation of deleterious mutations, the accumulation of non-synonymous relative to synonymous changes. And we see this at both the polymorphism level and also the divergence. So the oldest lineages are more heterozygous and have more evidence of deleterious mutation accumulation. So we see replicated, every transition is associated with relaxed selection, and the oldest ones are associated with more relaxed selection. And just to conclude, let's see if it's coming back. PowerPoint failed. Sex is good for your health.
Thanks, Stephen. We have time for a question?
Yep.
In the back, Steve.
Earlier in your talk, you pointed out that you had this selfing individual had conserved regions of heterozygosity or variation. Do you see any signs of genetic interaction between those sites or segregation distortion?
That's a good question. So certainly those regions have high levels of linkage disequilibrium and elevated polymorphism. We don't know if they, I'm not sure. We should have some data that would be relevant, but that's a good question. We should check.
Hi. So in a number of different species following whole genome duplication, there are particular genes that have been conserved across these different species as single gene copies. And I was wondering if you've looked at that set specifically through your studies.
Yeah, absolutely. So again, we're at a very early phase with this tetraploid, for example. But even there, we see more inactivating mutations, so it's more at the polymorphism level than gene loss. More of those in the same genes that tend to go down to single copy. Interestingly, we also, so in our outcrossing diploid species, we see more inactivating mutations in the recessive form in those genes as well. So that sort of fits with the idea that this dosage balance sort of idea, genes that get maintained as duplicate are more dosage dependent, can translate even to the diploid populations.
One more question from John.
So I'm wondering if there's something unique about the biology of Oenothera that this sexual-asexual dimorphism has evolved so many times independently.
Yeah, I think that's an excellent question, and it seems like there must be. It's somewhat unclear what that is. There is one other quirky feature of Oenothera, even the sexuals don't have much evidence of recombination across most of their length. So that might make translocations kind of more free to evolve. And so they start accumulating in populations until you get to a point where they're permanently heterozygous and asexual. So it may be that there's a high rate of translocations, and that generates this process.
Is that it's a highly repetitive genome as well, or not?
So It's a intermediate size. It's about 600 megabase or so, and sort of a diverse array of kind of tandem repeats and TEs, but it's not the most repetitive.
Thanks again, Stephen.
All right, thanks a lot. And just because I didn't have my acknowledgments, but I would like to thank everyone at JGI in particular.